Hi, I'm Mark Yarchoan, and I'm a physician scientist at Johns Hopkins focused on hepatobiliary tumors and particularly fibrolamellar HCC. And today we'll be talking to you about our research program studying the abnormal metabolism of fibrolamellar cancer cells, and in particular, a clinical program bringing forward a glutamine antagonist for fibrolamellar HCC. Marina?
Hi, everyone. My name is Marina Baretti. I'm a medical oncologist, a GI cancer specialist with a special focus on fibrolamellar cancer, and I will be the person to run the clinical program that Dr. Yarchoan just mentioned, and we are extremely excited to discuss with you what is the hypothesis behind this trial and what are the next steps for this trial to be available to all our patients.
Let me give a little background on how this whole program came about. As a physician who treats patients with fibrolamellar HCC, one of the things that we have observed for a long time is that some patients with fibrolamellar tumors actually develop hyperammonemia, meaning that the level of ammonia in their blood is abnormally high. And actually, I do recommend to patients with fibrolamellar HCC who are treated elsewhere that at least once in a while, the level of ammonia should be checked in the blood because this is a real problem for a lot of our patients. And ammonia is sort of a waste product. So a lot of cells in the body make ammonia, and the liver is a really critical organ in getting rid of ammonia. Most patients with fibrolamellar HCC have pretty normal liver function. Their livers work pretty well because they're young people without cirrhosis. And so the fact that they are presenting with high ammonia was a tip-off that something is strange with the metabolism of fibrolamellar tumor cells. And so our idea is whenever cancer does something that's different from a normal cell, it's a potential opportunity to take advantage of a potential vulnerability that cancer cells might have. And so we've tried to study this in the laboratory by turning cells into fibrolamellar cells by using a technique called CRISPR to CRISPR in the fusion that drives fibrolamellar tumors, and trying to understand what is the effect of turning a cell into a fibrolamellar cell. What is that effect on the cell's metabolism? And what we found was that when we make this particular modification that turns a cell into a fibrolamellar cancer cell, the tumor cells gobble up an amino acid called glutamine, and they pump out something called glutamate, which is something downstream of glutamine. And so the fibrolamellar cells, it's a hardwired property of these cells that they gobble up glutamine. And they're actually quite dependent on glutamine to survive. If you get rid of glutamine, the tumor cells don't do well. And it's kind of a curious thing. Why do the cancer cells use so much glutamine? Some of this may be that they need glutamine in order to grow, to sort of feed their synthesis of DNA. But another side effect of gobbling up glutamine and making lots of ammonia and acid is that these waste products actually poison the immune cells within the tumor. And so it's a sort of 2-for-1. The tumor cells like the glutamine, and they also use this pathway to hide from the immune system and turn off the immune system in the tumor cells. And so we have really focused on this as a potential way to attack fibrolamellar tumors, studying the use of a glutamine antagonist, something that blocks glutamine, to see if we can use that both to turn on the immune system in the cancer and to starve the tumor cell of something that the tumor cell really needs. DAWN is a very old drug that's a glutamine antagonist. It was studied in several cancers decades ago, and unfortunately, it really didn't work. It led to a lot of side effects for patients, particularly a lot of diarrhea and other GI side effects, and it didn't really have the effect that was hoped for within tumor cells. But another group at Hopkins, right down the hall from us, developed a new drug that is based on DAWN, but gets DAWN directly into the cancer cells. And the drug is now called DRP-104. And so we started using this in our mouse models to see if it could control fibrolamellar tumors. And what we found is that DON, or this new version of DON, doesn't really work on non-fibrolamellar cancer cells, but works very specifically within fibrolamellar cells and actually is able to turn on the immune system within the tumors. And so it doesn't work in a petri dish, but it really works in the mice who have an intact immune system. And in particular, if you combine it with an immunotherapy drug, the effect appears to be synergistic. So the two drugs work together to really turn on the immune system. You never want to rely on just one model before bringing something forward into patients, but actually, as we've been doing all these experiments, there have been two other groups that have similarly found that fibrolamellar cancer cells have a very abnormal metabolism with more ammonia and more use of glutamine. And this is Praveen's lab at Cornell and Sandy Simon's lab at Rockefeller have really converged on the same pathway. So it's very rare that three scientists all come to the same thing at the same time, but it really gives us confidence that what we're seeing is a real finding. And so we're now in the process of taking these preclinical discoveries and trying to figure out how do we bring this forward for patients to actually try to improve patient care. So I'll turn it over to Marina to describe a clinical trial that we're putting together.
Yeah, thanks so much, Mark. As you can see from this preclinical data, we are extremely excited about this trial. In the trial, we will combine this glutamine antagonist drug, DRP-104, with the anti-PD-L1 Durvalumab. Before going to the trial, I do want to mention, as Mark was saying, that this is a sort of second generation of glutamine antagonist. And this is not going to be the first time that we test this in a human or in patients more in general, because there is already a phase 1 trial for DRP-104 that has been conducted at different increasing dose, and overall really show that this is a totally different drug from DON, much better tolerated, no treatment-related toxicity that led to any discontinuation or to any, serious side effect. So we are pretty happy to see the safety and tolerability in this drug. And in our trial, we will enroll patients with advanced stage of fibrolamellar cancers. Patients of age 12 and above will be allowed in the trial. And because one of the hypotheses is that with this glutamine antagonist, you can really remodel and reprogram the tumor microenvironment to make them more sensitive to checkpoint inhibition, we are enrolling patients who had received prior checkpoint inhibitor therapy. The trial, as the two drugs, the DRP-104, is a subcutaneous injection that is taken twice a week in a continuous way. And then we have the Durvalumab, which is given at the standard dose for which Durvalumab has been already approved in other tumor types, including liver cancer, which is an IV infusion once every 28 days. So really, our overall hypothesis is one, that we can use this glutamine antagonist as a sort of a new metabolic checkpoint to turn on the efficacy of tumor immunotherapy in fibrolamellar cancer. Two, that combining DRP1 plus a checkpoint inhibitor can result in this robust FLC-specific T-cell response that, more importantly, can translate in a robust clinical benefit, shrinkage, and improvement of outcome for our patients. So to give you a little more specific on the eligibility criteria, as I mentioned, will be patients with advanced stages or not amenable of, curative intent surgery that had received at least one prior line of therapy with checkpoint inhibition, age 12 and above. And we have been pretty permissive in terms of the entry laboratory work in terms of both, hematological function and liver function, especially from the liver function perspective, because we do know that this is a liver disease, and often patients have already abnormal liver function, but that would not be an exclusion criteria for our trial. The only thing, of course, will be if you are not eligible for immune therapy for other reasons. Usually this is having, uncontrolled autoimmune disease or being post-transplant. At this time, unfortunately, we are not able to enroll because we have this combination with checkpoint inhibition. But of course, we want to be as inclusive as possible. So we are already thinking about potential, amendment in the future of the trial to also allow that special patient population.
So unfortunately, this trial right now is only available at Johns Hopkins. And again, that's in part because we're dealing with a brand new drug from, a company called Drayson Pharmaceuticals. And the access to this drug is, highly, highly controlled. But certainly if this trial starts to show success, our immediate goal will be to try to find other centers that could be added to the clinical trial to make this more available across the country. And in parallel, we're trying to work with the FDA and others to find ways that patients may be able to administer this drug at home outside of Johns Hopkins to minimize travel here. And we hope to have an update on that soon. We're really excited to, bring this opportunity, this clinical trial forward for patients. We know a lot of things don't work that look very promising in preclinical models, but I have to say that I'm really hopeful about this. And, I think the more therapies that we bring forward for our patients, the better, especially ones grounded in very rational and strong preclinical science. And we look forward to seeing the results and hope to have an update for you very soon.